Welcome to Section 3 of Cardiology. Throughout this section, I will be discussing the electrophysiology of the heart. In the first part of the section, we'll discuss action potentials, and in the second part, we will discuss EKGs. Let's get started. Before we jump into the details of cardiac electrophysiology, why don't we take a step back and look at some of the basic electrical anatomy. This is figure 2.3 from your text, which shows the normal anatomy of the cardiac electrical system. The major components of the system include the sinoatrial node, which you can see is near the opening of the superior vena cava within the right atrium, the atrioventricular node, the bundle of His, the right and left bundle branches, and the Purkinje fibers. For step 1, it's important to know where the anatomy of each component of the electrical system is. It's common for examiners to test you on the exact location of the bundle of His, the SA node, or other components of the system. We'll talk about this more in a minute, but each component of the electrical system has the ability to spontaneously depolarize. When this occurs, an electrical signal rapidly moves throughout the heart and is responsible for the coordinated fashion in which the heart contracts. So in the image here, we can see many of the arrows showing an electrical signal moving throughout the heart. It's important to know that the SA node normally sets the pace of the heart by spontaneously depolarizing faster than any other component of the electrical system. So the SA node is commonly called the pacemaker. The other components, such as the AV node or the bundle of His, also depolarize, but because they depolarize less frequently than the SA node, they ultimately become eclipsed by the SA node. For step 1, it's important to remember the order from fastest to slowest in which spontaneous depolarization occurs. The SA node depolarizes most frequently, and then the AV node, and then the bundle of His, and finally the Purkinje fibers. So let's write that out here. The SA node, the AV node, the bundle of His, and the Purkinje fibers, P for Purkinje. You'll be tested on this, so be sure to take a few moments and commit this to memory. Now let's take a moment to briefly talk about a similar topic, conduction velocity. Conduction velocity is the time required for an electrical signal to spread throughout cardiac tissue. So while the SA node spontaneously depolarizes more frequently than the Purkinje fibers, the electrical signal travels more rapidly in the Purkinje fibers than the SA node. For step 1, it's important to memorize which components of the system spontaneously depolarize fastest and which components have the fastest conduction velocity. So conduction velocity is fastest in the Purkinje fibers, and then the SA node, and then slowest in the AV node. In other words, the electrical signal travels about 1 to 4 meters per second in the Purkinje system, 1 meter per second in the SA node, and less than a meter per second in the AV node. Okay, moving on to action potentials. Much of the background to understanding action potentials is covered more in the chapter on neurology, so that's not the focus here. In this section, we'll be discussing action potentials in more detail and focusing on the 2 types of action potentials seen in the heart. As stated in your text, there are cardiac myocyte action potentials and pacemaker action potentials. Pacemaker action potentials occur in the SA node, AV node, bundle of His, and the Purkinje system, while cardiac myocyte action potentials occur in the other cells of the heart that don't comprise the electrical system. This would include the cells that make up, for example, the ventricles, the atria, and so forth. Let's take a second to see the big picture, and then we'll dive into the details of action potentials. This represents the heart, This is the sinoatrial node, the atrioventricular node, the bundle of His and the Purkinje system, and these will represent other cardiac cells, such as ventricular and atrial myocytes in green. Recall that the SA node receives input from the sympathetic nerves and the parasympathetic vagus nerves. Remember, these can alter the rate of spontaneous depolarization. So pacemaker action potentials occur throughout the electrical system, such as the SA node. So PM for pacemaker. And cardiac myocyte action potentials occur throughout other cardiac tissues, such as the ventricular myocytes. So CM for cardiac myocyte. Let's walk through how the electrical system works from start to finish. So the heart receives neural input to the SA node, and the SA node is constantly depolarizing on its own, but the rate can change depending on what the sympathetic and vagus nerves are telling it to do. Once depolarization occurs in the SA node, an electrical signal is generated, which moves throughout the electrical system. When the electrical signal travels through the cardiac myocytes, they depolarize and millions of synchronized cardiac myocyte action potentials occur. And this results in a coordinated cardiac muscle contraction. Ultimately, the electrical activity I've just described is what allows the heart to beat in a coordinated fashion and allows blood to move throughout the circulatory system. Okay, now let's compare and contrast the different action potentials. First, we'll focus on cardiac myocyte action potentials, and then we'll discuss pacemaker action potentials. This is figure 2.4 from your text, which shows a cardiac myocyte action potential. The y-axis is measured in millivolts, and the x-axis represents time. The numbers 0 through 4 represent the different phases of the action potential. The letter I next to the different phases stands for current, which is derived from Ohm's law. Remember, Ohm's law is I equals voltage over resistance, and the I stands for current. Sometimes on these action potential figures you may see ITO or ITI. The TO stands for transient outward, and the TI stands for transient inward. So for example, if you see IKTO channel, this just means there is a potassium channel, the K, that has a transient outward, the TO, current, the I. So the potassium channel has a transient outward current. In other words, an IKTO channel means the potassium channel allows potassium to move from the cytosol to the interstitium, which results in a change in voltage that you can see in an action potential. Okay, with this in mind, let's pull up a blank screen and go through each phase of the action potential. So this represents the cell membrane. This is the cytosol, and this is the interstitium. Recall that 3 ions play an important role in cardiac myocyte action potentials: sodium, potassium, and calcium. I'll also draw an action potential on the side as we go through each phase. Again, the y-axis is millivolts, and the x-axis is time. Remember from the chapter on general principles that an action potential is measured using a voltmeter, which is comprised of one electrode in the cytosol. We can draw that here. And another electrode in the interstitium. The difference in electrical potential between the two electrodes is called voltage, which is measured in millivolts. Okay, so when an electrical signal reaches the cardiac myocyte, the change in voltage causes the voltage-sensitive sodium channels to open, which allows sodium from the interstitium to enter the cell. As the positively charged sodium enters the cell, the voltage within the cell becomes more positive. And we can see this on the action potential. This part of the action potential is called phase 0. And it's sometimes referred to as the upstroke of the action potential. As the voltage within the cell becomes more positive, the voltage-sensitive potassium channels are opened, so potassium begins to leave the cell. Because positive charges are leaving the cell, the voltage starts to become more negative. This is called phase 1, or initial repolarization. Moments after the potassium channels open, the calcium channels open, and calcium begins to enter the cell. The combination of positively charged calcium entering the cell and positively charged potassium leaving the cell counteract each other, so the voltage during this part of the action potential stays relatively constant. So both potassium channels and calcium channels are opened during this phase. This is phase 2. Sometimes phase 2 is also referred to as the plateau phase of the action potential. Eventually, the calcium channels close while the potassium channels remain open. If only potassium is leaving the cell, then the loss of positive charges allows the cell to repolarize. This part of the action potential is called phase 3. Eventually, the voltage returns to normal, or returns to the resting membrane potential, which is called phase 4 of an action potential. Remember, the potassium channels are normally more permeable than other channels at rest, and this is responsible for the negative resting membrane potential. Okay, let's do an example. What drugs prolong phase 3 of the cardiac myocyte action potential? Before we answer the question, let's draw this out. Again, this is the cell membrane, the cytosol, and the interstitium. This is the graph again with millivolts in the y-axis and time in the x-axis. I'll also draw a normal cardiac myocyte action potential here. Again, the three ions are sodium, potassium, and calcium. Recall that potassium channels are responsible for phase 3 of an action potential. So potassium channel blocking drugs could prolong phase 3. Drugs that do this include ibutylide, amiodarone, sotalol, and dofetilide. So again, all of these drugs inhibit the potassium channel. And these drugs are commonly used to treat arrhythmias like atrial fibrillation and ventricular tachycardia. By decreasing the efflux of potassium, the cell takes longer to reach the resting membrane potential. So the new action potential will look something like this. As you can see, the slope of phase 3 has decreased as I've drawn here. Okay, moving on to pacemaker action potentials. This is figure 2.5 from your text, which shows a typical pacemaker action potential. The y-axis is measured in millivolts, and the x-axis is measured in time. As you can see, a pacemaker action potential looks quite different from a cardiac myocyte action potential. You'll notice that there are only 3 phases in pacemaker action potentials, and they're labeled in a way that may seem confusing. So we have phase 4, phase 0, and phase 3. Physiologists labeled pacemaker action potentials in a similar pattern that cardiac myocyte action potentials were labeled, but they were labeled after the labeling of cardiac myocyte action potentials had already been established. Because they noticed the initial repolarization and plateau phases were absent, they decided not to include phases 1 and 2. This is why we can see the odd labeling of phase 4, 0, and 3. You'll also notice in this figure that the ion channel responsible for phases 4 and 0 are actually different from the ion channels we saw in phases 4 and 0 in a cardiac myocyte action potential. Phase 4, or the resting membrane potential, is due to sodium and potassium funny channels rather than potassium leak channels. Phase 0, or the upstroke, is due to calcium channels rather than sodium channels. Finally, notice that phase 3 is the same in both types of action potentials, which is due to voltage-sensitive potassium channels. Okay, let's draw this out on the next slide. This is the cell membrane, this is the cytosol, and this is the interstitium. Again, I'll also draw the action potential on the side as we go through each phase. Remember, the y-axis is measured in millivolts, and the x-axis is measured in time. Again, the 3 ions in pacemaker action potentials are sodium, potassium, and calcium. One of the most important differences between pacemaker action potentials and cardiac myocyte action potentials is automaticity. In other words, pacemaker action potentials have the ability to spontaneously depolarize. They don't rely on neurotransmitters or nearby electrical signals for them to reach threshold. This unique ability is possible through the sodium-potassium funny channels. The idea here is that the cell has the ability to increase the permeability of sodium leak channels and decrease the permeability of potassium leak channels. You'll notice that both of these effects causes positive charges to accumulate in the cytosol, which means we'll notice an increase in voltage on our graph. As I mentioned before, this part of the action potential is called phase 4. So the voltage slowly starts to become more positive, and eventually the cell reaches threshold, which I'll draw right here. At this point, voltage-sensitive L-type calcium channels open, and calcium rapidly enters the cell. This rapid change in voltage can be seen on the graph here. Again, this is called phase 0 of the action potential. The calcium channels then close, and the potassium channels are open again. The loss of positively charged potassium from the cytosol to the interstitium causes the voltage to decrease. And this occurs until the cell reaches resting membrane potential, and the process starts all over again. So again, this is phase 3 of the action potential. Okay, with this in mind, let's do a question to solidify this material. Which phase or phases of the pacemaker action potential is altered by adenosine? Before we answer the question, let's draw this out. Again, this is the cell membrane, the cytosol, and the interstitium. And this is the graph with millivolts in the y-axis and time in the x-axis. I'll also draw a normal pacemaker action potential here. Recall that adenosine is an antiarrhythmic drug, and it works by increasing the efflux of potassium. So here's our potassium channel. Adenosine works by increasing the efflux of potassium. And it also works by decreasing the influx of calcium through L-type calcium channels. So calcium is entering the cell through L-type calcium channels, and adenosine blocks these channels. You can see that by increasing the efflux of potassium, that the resting membrane potential will decrease. So it will take longer for the cell to reach threshold. Let me draw this out. So instead of the resting membrane potential being right here, it'll drop to here. So phase 4 of the action potential will look something like this. By blocking the L-type calcium channels, we can see that the upstroke is also delayed because it takes longer for the same amount of calcium to enter the cell. So again, I'll draw a delayed upstroke. The potassium leak channels still function normally, so the repolarization phase is largely unaffected. So you can see phase 4 and phase 0 are both prolonged by adenosine. Okay, moving on to contraction and relaxation. I've included this into the electrophysiology section because I think it's helpful to see that contraction and relaxation are intricately connected to the electrical system. Let's talk about contraction first, and then we'll move on to relaxation. There are 5 major steps in contraction, which you can see on this slide. But before I read this to you, why don't we take a step back and draw this out on a blank slide, and then we'll see what your text says. So this is the cell membrane of a cardiac myocyte. This is the interstitium, and this is the cytosol. Recall that the SA node sends an electrical signal throughout the heart, which causes widespread depolarization. As the electrical signal reaches the cardiac myocyte, the cell senses the change in voltage, and the voltage-sensitive sodium channels open. This causes the cell to further depolarize and is the first step in contraction. The depolarization causes the voltage-sensitive L-type calcium channels to open, which allows calcium to enter the cell. This is the second step in contraction. Once in the cell, calcium binds to receptors on the sarcoplasmic reticulum called ryanodine receptors, which is the third step in contraction. So this is the sarcoplasmic reticulum. It has ryanodine receptors, and when calcium binds to these, that is the third step. It causes calcium within the sarcoplasmic reticulum to leave into the cytosol. This calcium can then bind to troponin C, which is the fourth step in contraction. This orange line I'm drawing in will represent tropomyosin, and the purple will represent actin. Remember that tropomyosin normally binds actin and prevents the myosin heads from binding to actin. So the important step here occurs when calcium binds to troponin C. When this occurs, the tropomyosin is released from actin. This allows myosin, so draw myosin here, to bind to actin, which can then cause muscle contraction. And this is the final step in contraction. Okay, with this picture drawn, let's do a few questions. Do you remember what marker is elevated in a patient with a myocardial infarction? Troponin I. Troponin I. It's not enough to remember troponin. You need to remember that troponin I is a very sensitive marker that indicates damage to the myocardium. Don't get this confused with troponin C, which I've drawn here. Okay, here's another question. What drugs block the L-type calcium channel that I've drawn here? Non-dihydropyridine calcium channel blockers. That's a class of drugs, and they include drugs like diltiazem and verapamil. So again, both of these drugs block the L-type calcium channels. Okay, one last note. It's important to remember that calcium enters both cardiac myocytes, which I've drawn here, and pacemaker cells through the L-type calcium channels. But only cardiac myocytes exhibit calcium-induced calcium release from the sarcoplasmic reticulum. In other words, cardiac pacemaker cells do not release calcium from the sarcoplasmic reticulum. So drugs like verapamil and diltiazem that block the L-type calcium channel have no effect on calcium-induced calcium release in pacemaker cells. Okay, let's see what your text says. So, voltage-gated sodium channels open. This causes the cell to depolarize, which then causes calcium to enter the cell through L-type calcium channels. This calcium then can bind to the ryanodine receptors on the sarcoplasmic reticulum, causing calcium from the sarcoplasmic reticulum to be released into the cytoplasm. This calcium then can bind troponin C, which causes tropomyosin to move away from actin, which then can allow the myosin heads to bind to actin. Okay, now let's discuss relaxation. Again, this is the cell membrane of a cardiac myocyte. This is the cytosol. This is the interstitium. We'll also draw the sarcoplasmic reticulum here. Recall that one of the final steps in contraction is calcium binding to troponin C. In order for the heart to relax, the cytosolic concentrations of calcium must decrease, so less calcium is bound to troponin C. This occurs through two mechanisms. First, the cytosolic calcium is pumped back into the sarcoplasmic reticulum through calcium ATPase channels, also known as SERCA. So I'll draw that here. And second, the calcium is pumped from the cytosol into the extracellular space through sodium-calcium antiporters. Remember, this pumps 3 sodium molecules into the cell in exchange for 2 calcium molecules. You can see that both of these mechanisms decrease the concentration of calcium in the cytosol. Now I'd like to focus on the circa channel because this is the most high yield. There's an important protein called phospholambin. And this protein inhibits circa. You can see that phospholambin would prevent cardiac relaxation by preventing cytosolic calcium from returning to the sarcoplasmic reticulum, which would obviously be problematic. The way the cell avoids this is through the enzyme protein kinase A, or pKa. pKa phosphorylates phospholambin. I'll just draw a P here to indicate that phospholambin has been phosphorylated. So when phospholambin is phosphorylated, it no longer inhibits circa. This means cytosolic calcium can enter the sarcoplasmic reticulum and the heart can relax. The degree of phosphorylated phospholambin to unphosphorylated phospholambin is the major determinant of how much calcium is pumped back into the SR, or sarcoplasmic reticulum. It's important to know that if more calcium is pumped into the SR during relaxation, then more calcium will be released from the SR during the next contraction. This is very high yield. If more calcium is available in the cytosol, then more calcium can bind to troponin C, which means the heart can contract with greater force. This is also referred to as contractility, which is also sometimes called inotropism. So remember, contractility is directly related to the concentration of intracellular calcium. Okay, one last thing about contraction and relaxation. Catecholamines like epinephrine and norepinephrine increase the activity of pKa. As I just showed, pKa will inhibit phospholambin, so more calcium can enter the sarcoplasmic reticulum during relaxation, which also means more calcium can be released from the SR during the next contraction. This makes sense. We'd expect the heart rate and the force of heart contraction to increase during the fight or flight response, and this is one of the mechanisms that explains this physiology. Okay, let's see what your text says. Cytosolic calcium is moved into the SR through the circa channel. Phospholambin normally inhibits circa, and protein kinase A phosphorylates phospholambin. When phospholambin is phosphorylated, it's unable to inhibit circa. Catecholamines upregulate protein kinase A, which ultimately inhibits phospholambin and allows the circa channel to pump more calcium back into the sarcoplasmic reticulum. The increased storage of calcium in the SR will result in a stronger subsequent contraction. Finally, calcium also moves from the cytosol to the extracellular space through the sodium-calcium antiporter. Okay, let's wrap this section up. In the next few slides, we'll talk about ionotropes, chronotropes, and dromotropes. These all sound similar and can be kind of confusing, so I'd like to take a second and define each of them. Inotropism, as we just discussed, refers to the ability of the heart to produce changes in contractility. Recall that contractility is directly related to the concentration of intracellular calcium. If more calcium is available, then more troponin C is able to move tropomyosin from the actin sites, which means more myosin can bind actin, resulting in a more forceful contraction. An example of a positive ionotrope would be sympathetic stimulation of the heart. Epinephrine and norepinephrine combine to the beta-1 receptors, which increases the inward calcium current by upregulating pKa, which we just talked about. Chronotropes alter heart rate, and they do this by regulating the influx of sodium in the sinoatrial nodal cells. Recall that in pacemaker cells, sodium is necessary to reach threshold. It shouldn't be too hard to understand that increasing the permeability of sodium channels will allow the cell to reach threshold more rapidly, which will then increase the heart rate. Examples of positive chronotropes include drugs like dopamine, catecholamines, and adrenergic agonists. Finally, dromotropes produce changes in conduction velocity, mainly in the AV node. This is done by regulating the influx of calcium. Let's draw this out. This is a pacemaker cell in the electrical system of the heart. This is the cytosol. This is the interstitium. I'll draw a graph here on the side to draw out an action potential. Remember that during phase 0, positively charged sodium and potassium ions are accumulating in the cell. Eventually, the voltage becomes more positive and the voltage-sensitive calcium channels open. Recall that calcium influx is responsible for the upstroke of a pacemaker action potential. So we have sodium and potassium that are accumulating in the cell, and once we reach threshold, the calcium channels open, and we have the upstroke. So a positive dromotrope will increase calcium influx, which will result in a more forceful upstroke. A dromotropic agent can increase the influx of calcium, which means more calcium enters the cytosol. The way I like to think about this is the massive influx of positive charges repels other positive charges in the cytosol. And the force in which these charges spread out increases. So you can see that the electrical signal propagates throughout the cell more rapidly, and this is why the conduction velocity increases. Can you think of any negative dromotropic drugs? Non-dihydropyridine calcium channel blockers, like verapamil and diltiazem. So in summary, inotropes alter intracellular calcium concentrations in cardiac myocytes, and chronotropes alter sodium influx in SA nodal cells, and finally, dromotropes alter calcium influx in the AV nodal cells. And that concludes this section.